Hello and welcome to today's video. It is on genes, alleles, gene linkage and variation. Being the last video for the term though, I thought I would wear my unicorn beanie because it's pretty cool. And it's actually from Nepal. My brother bought it for me when he was over there last year to do the Everest base camp trek. So, cool. Let's get into it. The learning goal for today is to talk about what we started covering last week, that being meiosis 1 and 2 and how various genetic processes result in genetic variation. There's a few key terms and areas that we did not so much go into last week, so I will be covering those with you today. You will find this in 8.3 of your textbook. There's a few key terms down the side here, which again I won't speak to directly, but you can follow along as we go about this video. So genes. Genes are a region of DNA made up of nucleotides, which encodes for a specific protein. And that protein has a specific function, be that involved in the immune system or coding for a specific physical or chemical trait. Genes code for proteins, and proteins are what make up you and I. The order of the nucleotides is what determines the gene. So the nucleotides vary, and therefore so do the genes. And chromosomes carry many different genes on each chromosome. In this image here, you can see that the ordering of these nucleotides results in different genes. Last week, I gave an example of there being a section of DNA, which is what a gene is, on chromosome 15 that is responsible for eye color. I would like to point out though that it's really not that simple, especially for complex traits such as eye color. It's generally a combination of many genes, even on different chromosomes, that result in a particular physical trait. So that's just a little clarification point. It's a simplified example of how genetics works. So welcome to genetics. Genes that are close together and on the same chromosome are said to be linked. So these genes are together on the same chromosome. We could call this gene linkage. Another key term which I introduced last week was alleles. So an allele is a variation of the same gene. It is based on equivalent positions on homologous chromosomes, i.e. if you've got one from mum and one from dad, we can have the same gene on those chromosomes. So here's the gene that results in the color of the flower. That's what we're looking at here. And say if this was the gene from mum, here is the gene for color, but it varies between mum and dad, so therefore it's a different variation of the same gene, and that is therefore what we call an allele. Although there may be many different versions of alleles, so in this example, many different, say, flower colors, an individual can only carry two versions. So that would be one from mum and one from dad. In this diagram, we are looking at the green pea and the gene responsible for causing the pea to either be round or wrinkled. There are two variations to this gene, represented by big R and little r. And it takes both of these alleles to code for the final physical trait. And depending on the combination of both of these alleles, the pea is either wrinkled or round. Before we discuss what we are looking at here, let me clarify a few more genetic terms that will help us understand this. The first is genotype. And genotype refers to the genetic makeup. So in this case, for this gene, we use the symbols uppercase r and lowercase r to represent the two alleles, the genotype. The second term I would like to discuss is phenotype. And the phenotype is the physical expression of the genotype. So in this case, that physical expression is either round or wrinkled. There's a couple more key terms that I will introduce now. And the first one is dominant allele, and the second one is recessive allele. So dominant refers to when an allele for a gene overrides the effects of the recessive allele. And recessive refers to when the dominant gene is taking over effect, while recessive is the effect, the gene that is being masked by the dominant gene. The dominant allele is always represented by an uppercase letter, be whatever that it may be, and the recessive by the lowercase version of that letter. So in this case, the uppercase R represents the round version of the allele. That is the dominant allele, and the lowercase r represents the wrinkled version. A couple more key terms. Another key term is homozygous and the other heterozygous. So homozygous refers to when both alleles are the same, as the word homo suggests, of the same or one. Whereas heterozygous refers to when the alleles are both different. So in this case, homozygous may either be uppercase RR or lowercase RR. These are the allele combinations we are talking about. So we're talking about the genotype, and what that genotype physically results in would therefore be the phenotype. So let's have a look at this diagram now using our knowledge of these different terms. Homozygous, we have two versions. We have uppercase R and lowercase r. Then we have two heterozygous outcomes here. They are the same, uppercase R and lowercase r. What this diagram is representing, and we're not going to go into great detail here, is that we have a parent over this side and a parent over this side. Both these parents in this case are heterozygous, that being they have an uppercase and a lowercase r. This squared is called a Punnett square, shows us the possible outcomes of the offspring based on their genotype. So if this parent with an uppercase R and lowercase r gave one of their uppercase R and received another uppercase R from the other parent, then one possible option of their offspring, their genotype, would be homozygous uppercase RR. They could also contribute a lowercase r, and so the other parent could contribute an uppercase R, so therefore we would again have a round P because the uppercase R is dominant for a round seed, resulting in these two options. But then if both contributed their lowercase r, we could also have a wrinkled P with homozygous little r as the result. In this case, you can see the dominant trait, the common physical trait expressed in the phenotype of the P, is round. We are looking at the P in this case to discuss these terms as the genotype results in an obvious physical trait, phenotype. And this is a great example of how genetics works. We're not going into the area of Mendelian genetics so much in this video. That's at a time later to come, where we will look in more depth at Punnett squares, which is what this is. This video is basically an introduction and an overview to a few key terms and an introduction to Mendelian genetics, which is what we are looking at here nonetheless. And this was developed, this whole understanding in Mendelian genetics, by this man. He was an Austrian Austrian monk, botanist, and teacher, and he is now known as the father of genetics. His name is Gregor Mendel. He developed what is called the law of segregation, which is again what we're talking about here. It states that genes occur in pairs, one from mum and one from dad. The law of segregation, meaning they split. He also developed what is called the law of independent assortment, and this simply states that allele pairs segregate, split, and assort independently of each other. Just like we're seeing here, they split and they can assort independently, resulting in all these possible outcomes. So that is all from 8.3 of the text. The next section of your text, and finishing up the content of this term and of chapter 8, you can find in 8.4. I will not talk to this in the video here, but what you can see and get the picture of is that it's to do with genetic engineering. Here is a quote by a man called Edward Teller. He was a Hungarian-American theoretical physicist who is colloquially known as the father of the hydrogen bomb. And he gave this cool quote. He says, "The science of today is the technology of tomorrow." So this science to do with the genome, genes, is the science of today and decades now. But we are starting to see this quote come into fruition, that it is becoming the science, the technology of tomorrow. So check out the bio description here in this video for a couple of articles. One is on IVF and another is on CRISPR. This is the cutting edge technology that is happening in the biology world at the moment. The quote that I will leave you with for today, though, is from a man called Francis Collins. He is an author, he is also a Christian, and he's a scientist who has been attributed and credited with cracking the genome. And he said this: "I believe God did intend in giving us intelligence, to give us the opportunity to investigate and appreciate the wonders of his creation. He is not threatened by our scientific adventures." Pretty cool, hey? Well, guys, bless you. I hope that this little video was informative and intriguing, and I look forward to seeing you in class.